So it's my pleasure now to introduce the third plenary speaker of this morning's session. That's Janaina Mourao-Miranda. Now, let me just tell you a little bit about Janaina. Janaina is a professor and a Wellcome Trust Senior Research Fellow at the Computer Science Department in the University College London. And I just want to say that I've known Janaina now for about 11, 12 years. We've had a couple of collaborations. And it's been a fascinating journey for me to kind of learn about the computational neuroscience framework that Janaina brings to psychiatry. Janaina is a bioengineer by trade, and yet she has this uncanny ability to actually understand some of the really nuanced ideas that we as clinical scientists even struggle with. So, like Yael, Janaina sort of is almost like a hybrid scientist. She brings a computational approach to clinical neuroscience hard questions. She's been the recipient of 2 incredibly prestigious awards from the Wellcome Trust. For those of you not so familiar with the UK funding structure, the Wellcome Trust is probably the largest non-governmental funding body in the UK and indeed in Europe, but it awards very infrequently. And Janaina has been particularly good and productive in receiving not only a Wellcome Trust Career Development Fellowship, but also a highly prestigious and coveted Wellcome Trust Senior Research Fellowship. And she's in the Computer Science Department at UCL. So today, Janaina is going to be telling us about her more recent work where she's been developing and applying machine learning techniques to neuroimaging data in psychiatric and healthy populations. And really what Janaina's been, her mission of her lab has been to use these more sophisticated machine learning techniques with neuroimaging data to kind of answer very fundamental clinically relevant questions, diagnostic classification, risk prediction, etc. So it's my pleasure today to introduce Janaina and her talk, What Can Machine Learning and Neuroimaging Techniques Bring to Psychiatry? Janaina.
So, thank you very much, Mary, for the very nice introduction, and thanks, Mary and Scott and the organizers for the invitations to be here to talk about my work. It's a great pleasure and honor. So, Mary already introduced the title of my talk today, and I will start just mentioning that I have no disclosures. So, in the talk today, I will use examples from the work I have done with clinical collaborators over the last years to illustrate how the combination of machine learning and neuroimage techniques can help us to address different questions and challenges in psychiatry, such as identifying biomarkers, improving diagnosis and prognosis of mental health disorders, and providing new tools to precision psychiatry. Let's start with neuroimage techniques. Neuroimage techniques have been around for almost 30 years now, and there is really a variety of techniques available that provide very good and detailed information about brain function, structure, and connectivity. And because of that, they hold enormous potentials as biomarkers for psychiatric and mental health disorders. But despite the fact that many studies, clinical neuroimage studies, have used different techniques to identify abnormalities, structural and functional abnormalities in different patient groups, so far there has been limited clinical translation. So I think an obvious question for us to ask is, why haven't we found neuroimage biomarkers yet? Or why, in other words, why neuroimage hasn't been translated to the clinical yet? And well, I think there are many different reasons for that. But today, I would like to focus on 2 challenges that I believe machine learning might help us to overcome. One challenge is the fact that when we talk about psychiatric and mental health disorders, and I'm sure you know better than me, the changes are very subtle and distributed in the brain. A situation that is different from many neurological disorders, for example, like dementia and Parkinson, that at least in some stage of the disease, you expect to see some localized structural changes in the brain. I think in psychiatry, there is a lot of data showing that the situation is much more challenging. So that is one challenge. The other challenge is that so far, most of the clinical neuroimage studies have focused on identifying group differences. So locating on the brain where are the differences between two groups, like patients and health controls, instead of focusing on individual prediction. So I would argue that in the search for neuroimage biomarkers, the question we would like to answer is how can we identify subtle patterns in neuroimage data that's useful for psychiatry diagnosis? And if we highlight the words neuroimage data and psychiatry diagnosis, we can actually replace these words and transform this question in a much more general question, which is how can we identify subtle patterns in complex data that is useful for prediction? And interesting, that's exactly one of the questions that machine learning tries to answer. And I will give a very brief introduction about machine learning. I believe most of people here will be familiar with machine learning at some level, but just a classical introduction or a classical definition is that machine learning is a subfield of computer science that gives computers the ability to learn without being explicitly programmed. And there are different types of learning in machine learning, and I will just give some intuition about two, which I think are the most useful, used types of learning. Perhaps the most known and applied framework in machine learning is the supervised learning framework. And here I'm just giving an example of this framework. So the idea with the supervised learning framework is that we train, we give examples, patterns, and labels. And in this example, I have handwritten digits, and I have the labels are the real digit of the number. And we give this as input, you know, the pairs of patterns and labels as input to a machine learning algorithm. So the algorithm during the training will learn the associations between the patterns and the labels. The outcome of the learning is a predictive function, which you can use during a testing phase to make predictions to new examples. And this framework has been applied to many different areas that I will mention later, but that is one type of learning, machine learning. There is another type of learning, which is unsupervised learning. And here in unsupervised learning, you know, the goal is that to give unlabeled examples as input to the machine learning algorithm. And you know, the aim is that the algorithm will find some underlying structure in this data that will enable us to do some tasks such as clustering the data into different groups. Can also be used for dimensionality reduction. But what I like to mention is that unsupervised learning is a more challenging task than supervised learning because it's very unconstrained. So here we don't have labels, and it's not very clear the task. Clustering is a very common task that is quite difficult to optimize. Here in this example, I arbitrarily split the numbers, the patterns, into the real digits that they belong to. But clusters depends on metrics and similarities, and I could have cluster things in a different way, like the bold numbers versus the non-bold numbers, or perhaps the ones formed by lines and the ones formed by curves. The point I want to make is that depending on the criteria, you might end up with different clusters and a number of clusters as well, which makes unsupervised learning a more difficult challenge to solve. And there are other types of learning, semi-supervised and reinforcement learning that I will not cover here because have not been much applied in clinical neuroimage data. So in terms of applications, we can find applications of machine learning nowadays everywhere. Just the example I give with digit recognition, with object recognition, face recognition, finance, advertising, genes, like recommendation engines, and neuroimage data analysis as well. In the context of neuroimage data analysis, the machine learning framework most commonly applied has been the supervised classification framework, which is a framework that I've just introduced. You know, and here in this context, you know, in the context of neuroimage, the goal is that we give examples of patterns and labels, but now the patterns are brain images, and the labels can be, for example, patients and healthy controls, and we give these examples as input to a machine learning algorithm. And as I mentioned before, during the train, we will learn, you know, how to discriminate the two patterns, and then the outcome is a predictive function, which we can then be used to make predictions when we have an image of a new subject. And there are very important advantages with this framework that make them very attractive to neuroimage data. One is the fact that it's a multivariate framework. So it does, when during the training, really the algorithm will look for the whole pattern that we give as input. If we give the whole brain pattern, it will look for the whole brain pattern, trying to see where are the differences between the groups. So it does take into account the distributed and subtle information in the image. And the second advantage is that once we train the model, we can use it to make individual subject predictions. So we can take this predictive function and apply it to data from a new subject. And actually, machine learning is all about predicting new subjects, new examples, and generalization. So then we can measure the performance of this model in the data that was not used to train the model. And obviously, I think it's the early applications of this approach to neuroimage were actually more in neuroscience as brain decoding or to classify brain states or fMRI tasks. But it didn't take long to people to realize that this had an important clinical potential. And basically over the last 10 years, this framework has been applied to address many different clinical questions in psychiatry, such as diagnosing across disease, predicting disease outcome, identifying at-risk subjects, and identifying treatment responders. And I truly believe that there is a large body of evidence already showing that there is predictive information in the neuroimage data. However, these studies have also been subject to some criticism. And the main criticism has been in terms of reproducibility. And I just want to show one figure from Gael Veracruz paper that I think summarize really well like the last 10 years of application of neuroimage-based classifications to different diagnoses, to diagnose of different disorders. So what we see here, what he did here, he did a meta-analysis of reviews of different classification diagnosis approaches. So each figure here is a different review, and each point represents a different study. And there are different diagnoses like Alzheimer's, depression, schizophrenia, psychosis, autism. But we see an overall trend here. So in one axis here, we have the reported accuracy. Let's see if I can. Here we have reported accuracy, and here we have sample size. And the overall trend we can see is that smaller sample size studies that use a smaller sample size tend to have higher accuracy than the ones with larger sample size. And although overall, all of them are able to make predictions, so there is, I don't want to be pessimistic, all of them, they are able to predict, to make predictions and make diagnoses, but we have this strange effect that when the sample size increases, the accuracies decrease, which is particularly strange and unpredictable, because in other machine learning applications to other fields, what we tend to see is that when the sample size increases, the accuracy increases. So there is a lot of, in the paper, he discusses many reasons for this issue, including, bias with small sample sizes, publication bias, cross-validation, different type of cross validation one uses, but I think one problem is the fact that larger samples are more difficult to control and they are more heterogeneous. And that is an important problem here with this application. And after having worked many years with neuroimage-based classifiers for psychiatric disorders, and I started realizing that there was an important limitation with this framework. And in my view, the main limitation with this framework is the fact that the clinical assessment that is quite rich for a patient is summarized into a single label, which is the categorical classification. And as you know, the categorical classification has been subject to a lot of criticism in terms of not aligning with findings from neuroscience, genetics, and not being able to predict treatment response. So when we use the categorical classifications to train these neuroimage-based classification models, that's our assumption, that the two groups that we want to classify, two or more, you can have multimodal, multi-class as well, problem, but the main assumption is that the two groups you want to classify are coming from two distributions, and these distributions are homogeneous, and we are able to find a boundary between them. But the real world is much more like that. So there is a lot of heterogeneity in the patient group and also in the healthy controls. So over the last years, my group have been trying to think about alternative machine learning frameworks that we can use, moving away from the categorical classification, but is still able to address clinical challenges with clinical neuroimage data. And I will show now a few examples of frameworks that we can use to do that. So one example is normative models. And normative models actually are very common in the medical areas. So the idea is that we train this normative model with measures from healthy controls or the general population, and then we can apply this model to new subjects to see if they have abnormal measures that might indicate at risk or a disease. And from a machine learning perspective, this problem can be framed as an outlier detection problem. And how it works with machine learning is that we train these algorithms, just giving an example from one class, which in machine learning we call the positive class, but here in terms of normative model would be the healthy controls. And then we train, you see here, we don't have labels, we just have this healthy group, and we train the outlier detection function with these subjects, and then once we finish the train, we can use it to classify other subjects. New subjects as outliers or no outliers. And an obvious application of this framework is to identify patients as outliers with respect to health controls. And a couple of years ago, I used this framework to test the hypothesis that we could treat the patient classification as an outlier detection problem. So in this study, what we did was we trained a machine learning algorithm that is called one-class support vector machine with data from health controls. And in the specific case, the data was patterns of fMRI to sad faces. And we trained this one-class SVM with the data from the health controls, and then we applied the model to a new group of health controls and a new group of depressed patients. And what we found was that 79% of the test controls were classified as non-outliers, but only about 0.5% of the patients were classified as outliers. So initially we thought, okay, this result is not very good because we expect an approach to have good sensitivity and specificity. But what we did was to look at the follow-up information of the patients. And some of the patients had responded to treatment and others not. And when we look at this follow-up information, we found that 70% of the responders were no outliers and 89% of the responders were outliers. So what the algorithm was telling us was that the real outliers were the subjects that did not respond to treatment. And we thought this was a very interesting result. And what we also did was we correlated the outlier score that we can have from the predictions with the depression score, and we find a very high and statistical significant correlation. So the more outliers the subjects were, the more depressed they were. So I think this is one example of how can we approach the problem without having to rely on the categorical classification. And another way that we can approach the problem is with regression models, which I think in psychiatry is called the dimensional approach. So the idea here is instead of trying to find neuroimage features that enable us to find two groups, to find a boundary between the two groups, we can now train a model that can predict clinical symptoms in a continuous way from these neuroimage measures. And hopefully, it can even cut across different categories, pretty much in line with ideas from the research domain criteria framework. So we train these models to predict symptoms in a transdiagnostic way. And a few years ago, before that, just to give the machine learning perspective on this model. So from a machine learning perspective, how we train these models is that we give the patterns and the labels, but here the labels are not categorical, as just continuous label. We give these patterns and labels as input to the machine learning algorithm, and we have these two phases, the training and the test phase. So once we train the model, we can use it to predict the clinical score or the behavior score for a new subject. And just to make a point here, this is quite different from the normal and regression models that normally people use to relate, the signal in one voxel to the clinical behavioral symptoms. Here we're really relating the whole brain pattern to a continuous clinical symptoms of behavior. This framework has been already applied to address different questions in psychiatry, such as predicting symptoms from brain scans and predicting personality traits from brain scans. And so a couple of years ago, we tried to test the hypothesis that we could use this framework to predict clinical symptoms that would cut across different categories. This study was a collaboration with Mary Phillips, and we used a data set that she had, and that was a data set of youth, young subjects between 12 and 17 years, and they had different diagnoses. But what they had in common was that all the subjects had some level of emotional and behavioral dysregulation. So what we did here, we trained a machine learning algorithm, a regression model, canal ridge regression model, to predict from patterns of brain activation to reward a measure that would capture their emotional and behavioral dysregulation, which was the Parent General Behavioral Inventory, the PGBI, and we were able to predict this score at 2 different time points from the patterns of brain activations to reward. So it was a very important proof of concept that for us that it would work. You know, we could predict in a transdiagnostic sample. So far, but one thing I haven't mentioned and I haven't touched so far is on the predictive function. So here we use a canal ridge regression model, which is a linear model. And so far in the talk, I've been representing this predictive function not by a black box, but a blue box. But I think there is this intuition or this underlying assumption that machine learning is a black box, and it can be, but I just want to make a point that doesn't need to be. So if we use linear models, which I believe for the high dimensional data that we have and the small sample size are probably the best options for neuroimaging, we can visualize the predictive function. And here I'm showing the predictive function for this study. But even though we can realize it, because of the multivariate nature of the approach, they are not very easy to interpret. So what does they mean? When we look at this map, it represents the model parameters, you know, as a spatial representation of the predictive functions, and tell us and show us each voxel in the brain, which was its contribution for the prediction. However, because this is a multivariate approach, we cannot arbitrarily threshold, and we cannot think that the peaks, you know, the regions that have higher values, they have higher contribution, but the final prediction is a combination of all the voxels. Is a weighted combination of all the voxels, so all of them will contribute. So it's really, we cannot make local inference because the whole pattern is used for the prediction. So part of the work that we do in my group, in my lab, is try to improve the interpretability of these models. And I would just, there are different ways one can do that, using sparsity, feature selection, but I just want to show here one approach we've been used, which I think it has very good advantages for clinical applications, and we've been doing that by trying to embed prior knowledge about function and anatomy in the models. So far, I mentioned that we train these regression models with whole brain patterns and labels, but what we do, we developed an approach that we use an atlas, can be functional or structural atlas, to segment the whole brain patterns into regional patterns. So then we give this as input to a machine learning algorithm, and here in this case, the algorithm uses multiple kernel learning, which is an approach that has been developed in machine learning to combine different types of information for prediction. So the advantage of this approach is that it can learn the contribution of the different regions for predictions. So the outcome is also a predictive function, but it shows us, you know, the contribution of the different regions here. And this specifically algorithm that we are using has some sparsity constraints that I would just relate to give some intuition about these sparsity constraints is that the model will select a subset of most relevant regions for the prediction, pretty much like what the attention does, that we focus on what is more relevant. So here we use this sparsity, so the model can learn automatically during the training which are the regions that are most relevant for the prediction. And I will just show an example of a very recent application of this approach to also in a regression setting framework. This was also a collaboration with Mary Phillips, just published a few weeks ago. And what we did here, we applied this framework to predict bipolar risk factors in a group of distressed young adults. And I'll just be more specific here. We predicted maniac symptoms
from patterns of brain activations to reward expectancy. And here on the left side, what I'm showing here is the result is a scatter plot between the actual maniac symptoms and the predicted mania symptoms. And each point here represents a different subject, so the model does pretty well in predicting, you know, the maniac symptoms from patterns of brain activations to reward. But the different colors here represent the different diagnoses. We color code the subjects by the diagnosis just to emphasize the transdiagnostic nature of the study. So each color represents a different diagnosis, and some subjects have more than one, and some have no diagnosis. But none of them actually had bipolar disorder at the time. But we can see this, that it works. We can predict discontinuous symptoms in a transdiagnostic nature. And as I mentioned before, we use this algorithm that enables us to learn the contributions of the different regions for the predictions. And we found that, you know, the region with highest contribution for the prediction was the left ventral prefrontal cortex. So what we did then, we took this region as a prior in a second sample, and we used it in the second independent sample, just the patterns of brain activation within this region. And we were able to confirm the results that, and predict from the patterns of brain activation of these regions, we were able to predict in an independent sample the maniac symptoms. So this is just, I think the point I want to make is that we can use these models as well to learn something about the regions and the brain, even though they have their limitations. So, and I think these examples I've shown so far, show us how we can really use machine learning, moving away from the categorical classification. But in my view, they are still limited in the sense that all of them are based in a single neuroimaging modality, and we are just including, like, focusing one single clinical behavioral symptoms. So more recently, what my group has been trying to do is trying to add more information in the model. So we've been doing that using multiple source learning, which in machine learning is called, you know, multiple source learning. Sometimes it's called multi-view learning or data fusion. And the idea is here to combine multiple sources of information for prediction. And we can do this with two different goals. One is to combine these different sources of information to build multiple source predictive models, or we can combine these different sources of information to build multiple source models for stratification. And I will give some intuition about these two goals. Just to say here, I'm representing the different sources as different image modalities, but they can be genetics, can be any source of information. Behavioral, clinical. So with the first goal of building multiple source predictive models, so the framework basically is just an extension of the supervised framework I introduced before. Here we have, instead of just giving, you know, one type of image and the labels to the machine learning algorithm, we give here different types of, different modalities of images and the labels to train the machine learning algorithm. We can here use an algorithm exactly actually the same or very similar algorithm that we use to learn the different contributions of the regions, but here we use the multiple kernel learning, for example, to learn the different contributions of the different modalities, which can be very informative if you are looking for biomarkers. And then, of course, as I mentioned many times through the talk, once we train these models, given that we have multimodal information from a new subject, we can make predictions for new subjects. And this framework can even be extended to predict more than one clinical scores at the same time, using a framework that in machine learning is called multi-output prediction. And there is evidence from neuroimage data and from other applications that when you try to predict multiple scores that are correlated at the same time, it's more advantageous than trying to predict them individually. But now I want to spend the last time that I have actually on the second goal, which is building multiple source models for stratification. And here the idea is, again, to combine multiple sources of information as input to the machine learning algorithm, but much more in line with the unsupervised framework. The goal is that the algorithm will be able to find some common patterns across its different data sources that enable us to identify subgroups of subjects. pretty much in the line of, precision psychiatry, and the underlying, or the hypothesis, or the assumption would be that the subgroups might correspond to more homogeneous subgroups of patients that might be better target for treatment. And there are many different ways of, here is very abstract, I'm just giving as input to a machine learning algorithm. There are many different ways of approaching this problem. And I think the first thing we could think of is just we put everything in a clustering algorithm and we find the subgroups. But as I mentioned in the beginning of the talk, clustering is a very difficult problem to solve depending on the metrics and the similarities you use. You might have different solutions. It's difficult to know which the solution, maybe all solutions might be equally right in some way. So we've been, in my group, trying to find ways of approaching this problem, and I will now show an example how we've been approaching, but as I mentioned, there are different ways one can approach this problem. We've been approaching this problem trying to learn a latent space that captures associations across these different sources or this different type of data. And here I will just show an example of using this to learn a brain behavior latent space. So the idea is that we use models that are able to capture associations between two or more types of data. And in this example, I'm using brain data in one side and behavioral data on the other side. And here by behavioral data, I mean item levels from different questionnaires. So we use a model such as sparse partial least square, which is a version or an extension of partial least square that is able to work with very high dimensional data. And the way these models work is that it finds a latent space here, or weights for each of the data types. Here we have brain weights and behavior weights that capture association about, across these two data sources, across, in this case, brain and behavior. And because we impose a sparsity on the PLS solution, the approach will be able to select a subset, for example, of brain voxels that are associated with a subset of questionnaire items. So what we can do then, once we find these associations, we can project the brain data to compute brain scores for each subject, and we can do the same with the behavioral data to compute behavior scores. And what these scores tells us is how much each specific subject in our sample express this association. So finally, what we can do is that we plot each individual subject in this latent space, this brain behavior latent space, and we can see here how this association varies across our sample. And our hypothesis that if different subtypes of patients have different brain behavior associations, we can use this approach to identify the subgroups. And I will just finish showing an example of a very, very recent application of this approach. That to find a brain behavior latent space in healthy and depressed adolescents and young adults. Just to give some information about the sample use, we used here about 330 subjects from the Neuroscience and Psychiatry network. So subjects here, they had age between 14 and 24, and 10% of them had depression. So here we used the sparse partially squared to find associations between gray matter voxels and questionnaires, items from 16 different questionnaires that captured positive and negative aspects of mental health. What we did here also is that we added to the model 3 demographic variables as clinical behavior variables instead of removing or regressing out from the data. So what we found was that we found 2 associative effects. The first one was age-related effect, which, an association here, you see here, is because it's a sparse method, the only variable that was selected among 300 of items and some demographic information was age. So we found an association between age and prefrontal, temporal, and parietal and associative cortices, which is expected first considering the age range between 14 and 24. So that's most of the variance of the gray matter will be related to age. And is also pretty much in line with all the studies that show shrinkage of gray matter and cortical thickness during this age. And here what you can see in the bottom is the latent space for this first associative effect. This really is, the color code is age, and it's really capturing age effect on the brain. But we also were able to find a second associative effect that captured an associations between a subset of items, from the questionnaires related to depression, is self-harm, and a set of brain regions that included prefrontal, inferior temporal, and regions like insula and amygdala. So these are regions that have been implicated in depression and in a number of studies, functional and structure studies. So, and when we look here at the latent space for the second associative effect, we can see that really, in blue we have the healthy subjects, in red we have the depressed subjects, really capture these variations from healthy to depressed. And the point I want to make here is that it's a preliminary study. The sample is relatively small. The number of depressed patients is very small. But I think it's, for us, as a good proof of concept that we can use this framework to uncover, like, in a data-driven way, hidden dimensions of brain behavior associations that might be able to describe variations from normal to abnormal function. And in this case, we are going from healthy to depressed. We haven't done this yet, but something we can do is, okay, here, if this scores, this brain and behavior score related to depressed goes from healthy to depressed, so are people here at risk because they are in the middle? So we have still to look at the follow-up information, but we don't want to do too much on this because the sample here is relatively small for this type of analysis, but just to give some intuition about what we can get from this approach. So, and I want to finish actually with the vision of where we want to go and where we want to reach with this approach. So I think our goal is to apply this type of approach to much larger samples with much more variability and subgroups and different types of patients. And our hypothesis is that we will then be able to find a multidimensional brain behavior latent space. And there will be a region in this latent space where healthy people might lie, here represented by the blue people. And if you start expressing these associations too much, you might become at risk, represented here by the green people. And if you express too much of any or a combination of these associations, you might become a patient. And the hypothesis is that perhaps by using these dimensions to find subgroups of patients, we are more likely to find homogeneous groups that might be targeted for treatment. But that's the vision yet. We are not there yet. I want now to move on and finish to say that a lot of the work we do in the group is also to provide tools for the research community. And all the supervised framework, classification, and regression that I mentioned here is implemented in our toolbox, which is MATLAB-based, freely available. One can download from our website. So far, it has been focusing on neuroimaging data, like MRI type of data, but we are about to release the third version of the toolbox that will enable combine EEG, MEG, MRI data, and maybe even clinical behavior information into the models. And I want to finish by thanking my lab that support all this work and my colleagues and collaborators, and I'm happy to answer questions.
Thanks very much. We have time for a couple questions.
Dr. Miranda, thank you for an extremely balanced presentation. A comment and a question. Clearly, machine learning is an essential tool so that we can analyze large amounts of data efficiently, see patterns and relationships and structures that you might not otherwise see. But the question really is that the method itself is such that you generate hypotheses. You don't really test hypotheses. But the way I've seen almost all articles on machine learning is that it's never been presented that way. They're presented as, well, this predicts this, or that predicts this. And since it's hypothesis generating, essentially, would you agree that it's hypothesis generation, not hypothesis testing? And if, in fact, it's hypothesis generation, it's really important that results be actually replicated. And replication is something that's been virtually absent. There are new patterns, new studies, larger studies, different studies by the same group that generate new patterns, new relationships, but that same algorithm, if you will, is never exactly replicated. And a second thing that I would suggest is maybe, in terms of the machine learning folks are doing their job. They're developing their methodology, their techniques. I think psychiatry needs to do a better job of thinking about specific application. If it's a predictive function, exactly what are you predicting? And how do you test whether these predictions hold out or not? Similarly, if you're generating different groups, then exactly how they're different. And I harken back to Dr. McCarroll's presentation, for example, in which he spoke about machine learning generating that, when he looked at the interspecies comparison of the interneurons, that there was a subgroup that was distinct. There was a new subgroup, if you will, and then finding differences in terms of the neurotransmitter. So really taking that steps ahead, and I've not really seen that in much of machine learning in psychiatry.
No, I think I would agree with all the arguments. I think in terms of the first question, if it's hypothesis testing or generating, I think in general, machine learning is an exploratory approach. However, there are special cases. I think one can use it to test specific hypotheses. For example, if you have a very strong hypothesis of a region being predictive, you can go and use the patterns within this region to test a hypothesis, so you can frame it, but most of the applications are exploratory approach that I totally agree that, and I mentioned through the talk that the need for replication is fundamental, because a machine learning framework, it does a cross-validation. We always, for all the results I show here, is always results on the test. It's not the data that has been used to train the models. However, having said that, because of sample size are smaller, what people use are cross-validation. So you have all these loops that, data is used for training, and then it's used for test, and then you average everything. So it's not completely independent. So I agree that the way to go is to be able to demonstrate. Now you train an algorithm to predict something, you have to demonstrate that generalizes in a completely independent sample. Without doing that, is really difficult, because there are so many, something I haven't had time because it was not a technical question. I didn't want to go into the technical details, but there's so many things you can do wrong, like overfitting. There's so many, I haven't known a single person, including myself, that hasn't overfitted the data at some point. So if you are starting to work it, just take for granted that you're probably doing overfitting at some. So you have really to think a lot to exclude the overfitting. And that's a lot of the results that we see in papers are people who are overfitting. They do feature selection on the whole sample. It's a very common error, the whole subject. And then they split the subject by selecting the features. But that you should know. That's a classical example. But that's also causes a lot of these problems of difficulties to replicate. And there is another point of validating the results. Yeah, in terms of the unsupervised part, I think it's the most exciting, and there's a huge opportunity there for machine learning to help with patient stratification. On the other side, there is a big risk because, as I mentioned, you can cluster things in a different way, and you might actually find a story for all the different types of clustering algorithms. You always find a story you can tell, but you have to validate this. You really need to validate and replicate, and there is, I think, a very recent paper, I won't go into the details, that show this stratification, and people have not been able to replicate. So, yeah, I think these are all serious issues. So there is potential, but there is a big risk.
Hi, thank you for an excellent talk. I was curious to hear your thoughts on whether there's a systematic way, when you're using multimodal data from different modalities, to add and remove these iteratively to figure out whether they're capturing the same features and accounting for the same kind of variability across modalities, or if it's different, and if there's a systematic way to approach that.
Okay, I don't know if I heard completely, but you're asking if, when you combine different modalities, if it's possible to learn if it's the same features from the different modalities, for example, the same brain regions across the different modalities, or different brain regions is what you're saying?
Yeah, sorry, can you hear me a little bit better? I was curious about whether different modalities, for example, something like EEG and MRI, if we can disentangle whether those are capturing the same variability across subjects, for example, in these predictive models. Like, are they picking up on the same kinds of features?
I haven't done it yet, but I think it's doable to do. We are looking forward, actually, for people to use like some of the latest tools that we've been provided exactly for that. So if depend on the model you use, you can or not. So that's the question. It will depend. If you use linear models like multiple kernel learning, the one I mentioned, you were able to see the contribution of each modality, and within each modality we should be able to see the contribution of each region. Although in the EEG data would be each time windows, for example, depending on what you use as input to the model. But yeah, I think there are models that when I like to use multiple kernel learning because I think as a hierarchical model, so you can learn the contribution of the different modalities, but within each modality you can learn the contribution of the different groupings you might have being regions or time windows within each modality. So I think it's possible. There is a huge potential there. But on the other side, there are also models that are non-linear, very difficult, or really black box that you just know you predict, but you don't know from where it comes from.
Thank you.
We get time for two more, one back there and then one here.
Right. I have a similar question to the first one, but I'll ask it a different way. You use the word predict a lot, but typically in medicine, in diagnostic tests, you have a positive predictive value and a negative predictive value and a certain number that's a goal. Can you take your data and put it as a positive or negative predictive value? That is, if you have a positive signal, what's the likelihood that's a true positive, or you have a negative signal that's a true negative? And if you can't do that with your data, is there a goal? Are you trying to get 80% or 90% or 70%? Because unless you have a goal, it's very hard to know whether you have reproducibility or not.
Okay, I actually would like to thank you for the question, because I wanted to talk about the meaning of the word predictive or predictive across fields. And I didn't include in the talk because I didn't want to take too much talk, but to my time of, and just want to, in order to answer your question, I want to talk about the difference in nomenclature. You know, in machine learning, when you use the word predictive, in general has a different meaning from what I hear people talk in psychiatry. So I've seen talks here and from the papers I read in the word predictive. I'm not talking about positive predictive value and negative predictive value, but just the word predictive in general in psychiatry is used to say, okay, if you have a variable and you find an association with some variable set of variables, you find association with a future outcome, and you say, okay, this association shows me that this set of variables are predictive of outcome. So that's the usual meaning that I hear. So it's about to refer about something in the future in general. Machine learning, the word predictive has a different meaning in the field. So it means, when you talk about predictive, it means that you train the model to find a relationship between patterns and a label, and you're able to predict the labels. Can be labels from the future, because if you train the machine learning to predict outcome, it can be just the symptoms now. So the word doesn't refer necessarily. So the word predictive, it means that we are predicting independent data. So we train it, it means that we are able to make predictions in independent data. And it's what we presented in the papers, what we predict in independent data. So it's a different meaning. But having said that, from all these models, you have a prediction for each subject, and you can compute with the standard equations like the positive predictive value, the negative predictive value, and they should be on the papers. Like in machine learning, normally they have different names like recall, precision. Each field has different nomenclatures, but you can have these metrics. I haven't talked about them, but in the papers you can include for each of the model these more specific metrics.
Yeah, thanks for that clarification. I think it's important then that we understand the meaning of the terms as we use them, and I'll just make that case.
All right, one quick one.
Yeah, thank you very much for a great talk. And one of the things that we've had difficulties doing, and it is impossible, is predicting the future. So maybe we should start predicting the past. Might be a good idea. But thinking through the clinical or the behavioral parameters or things that we're going to measure, I'm interested in your perspective on how we could potentially prioritize that. And then the second part of my question really relates to time and how the modality, if you will, of time will be factored in, and how can we do that in relationship to the behavioral measures that would then pull into in a multimodal way for imaging and either other physiological measures. So bringing it all together. What are your thoughts on integrating clinical time?
Very, very interesting question. So the first one was about how to select like tasks and behavior. I think it depends how strong is the hypothesis. If my approach, because I don't have very strong hypothesis for most of the data I work with, I tend to use sparse models, and in the example I give here. In the example with the adolescent data from the NSPN network, we just had on the behavioral clinical information, we had 16 different questionnaires which we end up, and we use the items of the questionnaires, and we let the model tell us which of these items are linked to the brain. Because if they're not linked to the brain, if they, and the model, like the one I showed, really saw what is, across the sample, what is that when I vary this behavioral clinical measure, and we find which ones vary together with the brain, so they are mapped into the brain. So usually my approach is to be more exploratory and let sparse models to choose it. And I haven't had time to go into the details. Everything we have to validate in a holdout sample, and we have all these cross-validation approaches and different frameworks to make sure we are finding something that generalizes beyond our sample. That's the first question, but I don't think it's the only way to be exploratory. I think if you have a strong hypothesis, maybe it's better focus on behaviors and questions and tasks that make sense for a specific, very specific clinical question. The second is how to include time. I think that's the next step, to make these models to account for longitudinal information. There has not been a lot of development, but there are approaches in machine learning, like they call multitask learning, which has a completely different meaning for multitask, but the idea is that you can combine data. Normally it's used if you want to make predictions from 3 different schools, and you treat each school as a task, and you learn a model that learns a different school, like treat each of the data as a different task, but knows that there is something similar across the 3 of the different tasks, and then can predict performance for 3 different schools using this multitask model. You can probably translate that to these type of things. We have 3 different time points, each time point representing a task or a model, and you have this hierarchy that, maybe different time points will contribute, but that's just an idea that, but I think the next step is probably to think about how we can embed temporal information into the models.